tonight, a very big subject. And so, ladies and gentlemen, we now have the bittersweet pleasure of welcoming our final speaker to the stage. So let's give the biggest applause that you've done so far to James Ward Taylor!
Good evening, everybody. My name's James. I'm doing a PhD in chemistry here at the University of Glasgow. And I'm going to start off with a few misconceptions about science that really grind my gears. The first one is this stupid stereotype that scientists are never getting laid. I mean, I'm not. But I'm sure plenty of other ones are. The second big misconception that I'm going to debunk today is that doing public engagement in science is glamorous. And I thought that, but then I helped out in a documentary that's being filmed in our lab, and I learned that being in a science documentary is a bit like being in porn. It feels really good, but no one wants to see your face. The third big misconception about science that annoys me is that making a molecule is like putting together molecular Lego or Connects. And I thought that, but then I went and studied chemistry and I found out that I'd been lied to. And that making a molecule is not like Lego or Connects at all. It's a lot more like those foam letters that you get in the bath. So if you want to make a molecule, right, you want to do a chemical reaction, imagine this. You get a bucket full of those foam letters, right? Chuck them into the bath. Throw in a rubber duck, because you can't have a bath without a rubber duck. Maybe one of them glittery bath bombs as well, when you're anything like me. Don't judge. And then you get in there, and you thrash around like some crazed whale. And then you get out, maybe put some clothes on. And you get your colander and you sift through the bath. And you sieve out all the letters and just hope that they've all magically rearranged themselves to form your chemical Hamlet. It's a lot more likely that you'll end up with molecular twilight. And I've been at this stuff for quite a while now. I've been trying to make this molecule, it's one tiny little molecule, just a smidgen of it, for 6 months. 6 months I've been in the lab and I haven't had any. And I've not managed to make any of this molecule either. The kinds of molecules I'm making are called amphiphiles. And an amphiphile is a bit like a molecular Glaswegian. It's got parts of it that are like water and parts of it that are like grease. And these molecular Glaswegians arrange themselves with grease in the middle and water on the outside. A bit like a load of people gathered around the munchie box on Socky Hall Street in the rain. If you don't know what a munchie box is, you go to the takeaway and they just give you a pizza box full of random shit they've got that's greasy. You've got pakora, you've got doner meat, maybe some chicken wings in there, and they'll throw in some Glasgow salad, just for good measure. Well, in chemistry, these assemblies are called micelles. And micelles are very tiny chemical compartments, many times smaller than any living cell. Well, I'm interested in the origin of life, the chemistry around minimal life. I don't just mean looking at my own sperm under the microscope. There are two kinds of scientists who use microscopes. The ones that have used them to look at their own sperm. And women. Well, I want to make self-replicating molecular Glaswegians. I don't mean just looking into the toilets at sleazies. In chemistry, you can design a molecule to fulfill a certain function. And there are certain things about molecular self-replicators that are similar with humans that might help you. For instance, self-replicating molecules, like humans, have recognition sites. You know what I mean? Bits that fit into other bits. And when these recognition sites just match up, the bits fit into the other bits perfectly, we call this complementarity, and it's the core concept of the system. Another similarity between humans and self-replicating molecules is that, well, molecules come in all different shapes and sizes. And sometimes you've got two molecules coming together, they're almost ready to bind, they're really close, and one of them's just too floppy. And it needs to spend time and energy reorganizing itself to get itself ready to go. But if you want your molecule to self-replicate, you want your molecule to be ready to go straight away. So you can design your molecule to be rigid from the get-go. And this is a concept called pre-organization. So if you want to start looking at the diagram and imagining these as people, pick your favorite gender, because we've got two of the same. And we're going top to tail. This is a 69, if it were. So another problem that you might encounter with both humans and self-replicating molecules, you've got one really tall person and one really short person, and neither of them are particularly flexible. Well, then you're going to have less than optimal binding. And that's not what you want in a molecular self-replicator. So you want your recognition sites to both be the same distance from each other. Now, that was the normal bit. Here's where it starts to get a little bit kinky. You see, in molecular self-replication, you start off with sort of just half a person binding first. So you get like a pair of legs getting in on the action first. I know, it's a bit fucked up. And then the torso comes in there. And then they're perfectly placed for chemistry to join them together. And then they can come apart and form new assemblies afterwards. So you start off with one of these, now you've got two. You can get 4, 8, and so on. But one of the problems you might have is these two things just stick together a bit too well. It's like the two people like each other too much, and they don't go and form other assemblies with any other molecules. And this is a bit of a shame, because then your rate of formation is limited by the rate at which the couples have had enough with each other. And this is a concept called template inhibition. So now we've got our design rules for designing self-replicating molecules. We've got our self-replicating molecule. We want to turn it into a molecular Glaswegian. So we add a part that loves grease to one side and add a part that loves water to the other and watch them join together. And then we test it to see now if you're screwed with the molecules that the self-replication orgy still happens. And it does. Fantastic. And now we want to put them into a reaction vessel and see if they do anything else that's interesting. So we're going to go to the crowded reaction vessel that is Socky Hall Street at 3 o'clock in the morning. So imagine this, you wander out of the club and there's a load of cool people about gathered around a munchie box. And everyone's getting on the munchie box, everyone's chatting away, having a good time, and people just keep joining the circle around the munchie box, getting in on the munchie box. The munchie box circle gets bigger and bigger, until eventually the guy in the middle tells everyone to fuck off and get their own munchie box. And then someone does, and starts a new circle. And now there are 2 munchie box circles. And this can go on and split into 4, 8, and so on, just like the division of living cells, which is why I find this fascinating. But there's the question of how did the first munchie micelle come to be in the first place? Can't have just started spontaneously from nothing on Socky Hall Street. Well, if you imagine this, you walk out the club and there's nobody there. It's pissing it down with rain, maybe you don't get food, maybe you do and you wander home with it. But if you come out of the club and there's a load of people having a good time, they're all chatting away, they're all standing around this munchie box, you might be tempted to join in the fun. Well, there's a number in chemistry that explains this. It's called the critical micelle concentration, or critical munchie box concentration. And it's a number of concentration of molecules below which you don't really see micelles. So it had to get started with someone coming out of the club, having a brainwave and thinking, I know what I need after 9 pints of warm lager just before getting into a taxi home. I'm going to clog my arteries. I'm going to get a munchie box. And this happens. This happens here. And it not only starts off the micelle formation process, but it continues to happen as all the other reactions happen. But it's a very slow background process, since humans have a very high activation barrier against soggy pizza. So, well, no matter what reaction pathway your chemicals follow, at the end of the night, the munchie boxes are all going to be stuck to the sides of bus shelters. And hundreds of people will wake up the next morning thinking, I know I got into one fucked up orgy last night. Why the hell did I have to eat that? Because I'm interested in observing the template self-replication orgy and the micelle munchie box mess all in the same reaction. That's what I'd like to see. And I've not found it yet, because I've had experimental difficulties. But I'm not just a random pervert who does chemistry. I've wrote a song about my experimental difficulties. It's a song about the fridge in my lab. Hello, lab fridge, my old friend. I've come to open you again. Amine bottle slowly leaking, toxic vapor softly seeping. When my nose was hit by the stench of a rotting whale, as I inhaled and smelled the smell of science. And in the fridge's light I saw 200 bottles, maybe more. If you don't label flasks of gunge, fuck you. If I don't find it soon, I think I'll spew. And I wonder why they all avoid me the next few days, 6 feet away, because I'll smell of science. Fool, said the fridge, you cannot know that carbon chains and micelles grow. Bonds are forming without templating, templates bind without dissociating. And the micelles are a mess of every shape and size, I theorize, and now you smell of science. Thank you very much, guys. Good night.